Started on slideshow, right? Okay. And let me just make a quick. If I could have your attention 1 second. Hi. Some of you may remember that a week ago Wednesday we were not here, we were elsewhere. MedAV, in fact, videotaped us and provided us with a WMV file. It's a 180 megabyte WMV file, so it's a different size than our others are, and it's a little different quality. But if you want to look at that video, it's available in the big format through the icon clicky thingy that you usually use, or off of Google through the Google links that are also usually there for it. Just be prepared for the fact that it's a little different in quality because it was actually a videotape. And now, Carissa.
Thank you so much. Okay, well, happy Friday, everybody. It's Friday the 13th, if you haven't noticed. So hopefully that doesn't bode well for the lecture. But so we're going to go ahead and start. Today we're moving on to chapter 18, which is talking about cancer and DNA repair. So it actually fits very well following DNA replication because there's a lot of interaction between the two processes of DNA replication and its relationship to cancer and how it leads to the development of cancer. So there's going to be some referral back to chapter 17, so maybe it'll be a good review for you or help you brush up on things you might have missed from the last chapter. But nonetheless, it should hopefully integrate this information really very well, helping you understand the fundamental cause of cancer, as well as some important genes and proteins that play roles in the development of cancer. So cancer is incredibly widespread. I would guess that everyone in this room knows at least one other person that has been affected by cancer in their life. And as you might know, that cancer actually comes in a wide variety of diseases. You can have cancer of nearly any organ that you might think of, and it also affects people in different ways as far as whether it's like external on the skin or whether it causes internal damage or pain or just variety of different symptoms as well as the types of diseases. So it's really actually very hard to sometimes get a grasp or a good feel for what cancer is. So we're going to look at some of the overall themes that relate to cancer, as well as narrow it down to just some specific genes and some specific proteins that are commonly known to be involved with a lot of different cancers. Fundamentally, though, cancer is a disease that results from problems that are found in DNA. So by problems, I mean inappropriate DNA replication or mistakes in the DNA, such as mutations, insertions, deletions, that sort of thing. And damage, DNA damage actually can affect the structure of DNA and thereby also lead to the development of cancer. And we're going to look a little bit at how damage affects DNA replication, as well as how mistakes might actually occur within the process of DNA replication as well. This picture is a picture of a breast cancer cell. Let me get this arrow going. There we go. All right. And you might take note that there's lots of these little spikies. I'm assuming that these are arteries supplying blood and nutrients to the tumor to help it grow even more. So before we move on too much into cancer, this whole chapter talks some about DNA repair, the mechanisms of repairing damage within the DNA. And the chapter actually starts with a great example of an organism that's really good at repairing DNA that's hurt or has lesions within it. And that's this little organism that's called Deinococcus radiodurans. And this is the most radio-resistant organism that is known on Earth. Just for comparison, in order to kill a human, you need an instantaneous exposure of 10 grays. And gray, abbreviated GY, is the measurement of radiation. So 10 grays exposure will kill a human. 50 grays will kill an entire culture of E. coli. In comparison, if you expose these Deinococcus to 5,000 grays of instantaneous radiation, it doesn't affect them at all. They continue living. In fact, up to 15,000 grays, there's a 37% survival rate. So they're very, very resistant to radiation, much more resistant than you or I would be to the same level of exposure. And this is an example of what's called an extremophile. So it's an organism that loves or survives well in extreme conditions. In fact, Deinococcus is a polyextremophile because not only does it survive radiation, it's also able to survive acid conditions, vacuum, lots of heat, or very cold conditions. So it loves very extreme conditions and survives well, while most other organisms would not survive in those sort of conditions. Deinococcus was first found by some food chemists, actually, who were looking for ways to sterilize food that was canned using radiation. And what happened was they found that they exposed the food within these cans to high levels of radiation. They thought high enough to kill any organism that might actually be within the food. But for some reason, it still turned up spoiled and would go bad. And it turned out that it's because Deinococcus was contaminating the food and resisting the radiation and thus causing the decay of the food. Deinococcus is actually now currently used to clean up some radioactive waste using some recombinant genes from other organisms that will actually clean up the waste. These organisms are resistant to any radiation that's around, so they're able to survive and accomplish this cleaning up. And it's really very useful as far as that goes. And the reason that Deinococcus is able to resist so much radiation is because of two facts. First of all, they have multiple copies of their genome within them. So say if one gene is hurt by the radiation, there's many other copies available to compensate for the damage. Also, they have extremely quick repair mechanisms for their DNA. So in response to radiation, a lot of times, radiation induces damage within the DNA, and Deinococcus is really good at immediately fixing that within a very short amount of time so that it doesn't have a very detrimental effect on the cell, and thus they're able to survive. And this just shows how important DNA repair actually is for an organism. And humans do have DNA repair mechanisms as well. We'll be talking about those later, possibly on Monday, but we're not quite as quick at it as Deinococcus and not quite as efficient as well. So it's really quite amazing organisms. So to move on to cancer. Now the question that everyone really wants to know is, what is cancer? And to be honest, it's hard to define. I took a semester course on cancer and causes of it, and I felt like I came out not really even knowing much more about it than when I went in, because it's such a complex disease. And it's because there's so many different types of cancer, and each one shows up in different ways. But basically what it comes down to is cancer is uncontrolled growth of cells. Also, it's cells that just, when they normally should die, and they have these signals for programmed cell death called apoptosis, they ignore these signals and they continue to grow, and they don't die when they should. And both of these, uncontrolled growth and failure to die, are linked to alterations in DNA. So something happens within genes to cause these cells to continue to grow. And there's an estimated 16,000 new cases of cancer that were discovered in Iowa alone in 2006. So it's extremely relevant to everyone. Excuse me. Yes? Right. Exactly. Yeah, so the question she asked was about cancer, if you couldn't hear. She was asking about, say, like smoking and how that might actually affect or lead to cancer. And she was asking, does that actually alter the DNA? And in fact, it does. We're going to look at a carcinogen from smoking in a little bit. So it does lead to damage in the DNA and alterations. So as you said, good segue, what leads to cancer? First of all, it's a combination of heredity as well as the environment that you're in. Your exposure to, say, smoking or sunlight, even some viruses or asbestos and nitrosamines, these are all DNA damaging agents which can contribute to the development of cancer. Additionally, on the hereditary side, individuals are born with genetic predispositions. So they might already have mutations within some of their genes which contribute to cancer development. For example, retinoblastoma is a developmental defect cancer with the eye that often shows up in children. Children that have one allele already mutated in this retinoblastoma gene are already genetically predisposed for the development of that cancer, and thus they develop cancer a lot earlier than some others who might not have this genetic predisposition. Basically, it boils down to that all these, both heredity and environment, contribute to genes that don't properly function. They can be overly active and do too much work, or they can be inactive or decreased in their ability to work. There's some common terms that are used for these types of genes, which you might have come across in the past. There's the proto-oncogene. Proto-oncogenes are normal genes that are found in the cell, and they're essential for cell growth and differentiation. They're good to have, but if they become activated or they're turned up in their activity in some way, this proto-oncogene can become an oncogene, which contributes to the development of the cell, often causing the cell to grow more and faster than it should. Also, there are tumor suppressor genes. This is also a normal gene in the cell that's necessary in the development of the cell and in the continued lifespan of the cell. But if this gene is lost, it can also contribute to carcinogen, because it might not be able to, if it's not active, then it doesn't help the cell stop dividing or doesn't promote the cell to go into apoptosis when it maybe should. So loss of a gene leading to cancer is a tumor suppressor gene. Increased activation of a normal gene leading to the development of cancer is an oncogene. And basically, oncogenes and tumor suppressor genes are the fundamental cause of cancer. Thus, cancer is a genetic disease. There's actually kind of a bit of a balance that has to occur between the tumor suppressor genes and proto-oncogenes within the cell in order for tumors to fully develop. In a normal cell, a proto-oncogene has relatively low activity. It's active, but not to the same extent that it might be in a cancer cell. So this bar graph represents the levels of activity for a proto-oncogene here in this brownish purple color, and in orange here would be the tumor suppressor gene. The tumor suppressor gene in a normal cell has relatively high activity. If you look at the activity of these same genes in a cancer cell, there's a possibility that this proto-oncogene may be activated. Thus, the activity would increase drastically, contributing to the cancer development. Another possibility is that the tumor suppressor gene would be inactivated and show less activation than it does in the normal cell. So it's possible that you might see actually both of these as well, where the proto-oncogene is greatly increased and the tumor suppressor gene is greatly decreased. It's a combination of many proto-oncogenes and tumor suppressor genes that can contribute to cancer development. Okay. And just to cover a few terms that are commonly used when referring to cancer, I'm sure this may be familiar to many of you, but just to make sure we're all on the same page. Carcinogenesis is the development of cancer. It's the process of going from a normal cell to a cancer cell. Transformation is the term that's used to refer to the act of converting a normal cell to a cancer cell. And a tumor is basically a big mass of cancer cells. So this picture here represents carcinogenesis and has many of these terms in it. Where you might see we start with a normal cell, and something such as a carcinogen initiates the process of carcinogenesis. As enough rounds of multiplication and say more damage is exposed to the cells, there's this genetic change transforming the cell into a malignant cell. And as that cell divides, it leads to development of a tumor, and thus cancer develops from normal cells that are mutated. So I've said, basically many times now, I've said that cancer is a genetic disease. It's a result of things happening within proto-oncogenes, tumor suppressor genes, leading to cancer. DNA damage can play a role in this process of carcinogenesis by introducing base substitutions or maybe deletions of the DNA, causing inappropriate modifications that aren't handled very well through the course of replication. There's many possibilities and many forms of DNA damage, and it's surprising how high of levels might be found within a cell with DNA damage. Also, as I mentioned, genes may be inherited from birth that contribute to carcinogenesis. So this little picture represents cells starting from birth, where a cell is introduced to, say, a carcinogen, say from smoking, introduces one mutation. That cell is actually likely to be okay. It's going to continue dividing and progressing, and the individual might not notice, might not have many ill effects from one mutation. But after, say, many repeated exposures to carcinogens or other environmental exposures, you can increase the number of mutations. Say another mutation is included in those cells, and that's propagated on, and then a third and a fourth, and so on, until enough important genes are mutated, inactivated, or overly activated, that it leads to transformation and tumor progression. Now, these genes, as you might expect, if many different mutations are required, there are several pathways in the cell that are regulatory that contribute to the normal cell cycle that are usually affected through the process of carcinogenesis. And this example is, this is an example of what's called the multiple hit theory of carcinogenesis. Just the idea that you need many hits of mutations before genes are cause transformation of the cells. Now, there's really no defined recipe for the progression of cancer. You can't sit down and say, well, first you need mutation in gene X, and then after that, mutation in gene Y has to happen, because it happens in many different orders and many different results, or many different causes for this. But there are some processes overall that we do know play roles in cancer development. Whether these processes are activated or inactivated depends on what it is. We're going to take a look at 5 different processes that participate in some way to cancer development. Now, the first one is a normal process of cell division. Mitogens are chemical messengers that are often sent from one cell to another, or from one cell to itself, to tell the cell that it's time to grow and to divide. In cancer cells, there's either an increased sensitivity to mitogens, or they produce many of their own mitogens, telling the cell that it needs to keep growing and keep dividing beyond the point where it normally should. And thus, these mitogens tell the signaling pathways that say that you need to grow. Basically, the signaling pathway is stuck on. So when maybe you should have a red light, instead you get a green light, and it stays on the green light, and the cell continues to grow and divide many, many times. Apoptosis is the process of programmed cell death. This is what happens, say, when the genes in a cell are damaged far enough that the cell says, maybe it's better just to package things up and quit, and not continue to divide, because that would be detrimental to the organism. Well, in cancer, many cancer cells are resistant to apoptosis signals. So again, when maybe the cell should be stopping, they don't sense the signal saying it's time to stop, and they continue to grow, and they don't die when they should. So apoptosis is not likely to occur with cancer cells. Instead, the cells continue to grow and develop into tumors. Another process that we've already learned a little bit about is telomere erosion. And this is commonly actually leads to apoptosis of cells as well. Because when telomeric DNA is shortened, and remember the telomeres are just additional DNA that's added onto the end of chromosomes to act as buffers, extensions of the chromosomes, so that replication doesn't degrade important genes in the chromosomes. Now normally, as the telomeres erode, as they're degraded from multiple rounds of replication, when they get close to the point where chromosomes might start being affected, the cell usually says it's time to die, time to quit dividing, because we might propagate inappropriately to other cells. But in cancer cells, actually, telomerase is activated, an increase in its activity, and thus it keeps telomeres longer, keeping the cells propagating and growing and dividing more than it should. So two other processes that are seen to play a role in cancer development are contact inhibition and anchorage dependence. Now this picture here shows an example of contact inhibition, where you can see these are normal cells that are growing. And usually what happens is the cell will grow to a certain point where it starts interacting with neighboring cells. And once they start reaching this point where they are close enough to each other, there's really no room for it to grow anymore. Normal cells usually stop growing at that point. But cancer cells, for some reason, don't seem to stop or sense when they're in close contact with other cells, and they continue to grow and to bulge out and crowd the whole area. So you can see in this picture with the cancer cells, they seem to continue to get basically just bulging out rather than reaching this confluent stage like normal cells would. Anchorage dependence refers to the fact that normal cells like to be anchored on something solid. Cancer cells, however, tend to be independent or not have anchorage dependence, and they can lift up and move from one site to another. Thus, in a tumor, if you have a tumor developing, cancer cells could actually lose their anchorage dependence, lift up, and leave and metastasize to another area in the body. That's why often you hear that cancer has spread when a person has had cancer and it spreads to another area in the body. That's the process of metastasis, where these cells pick up and move somewhere else and implant on another organ. And actually another factor in cancer that contributes to metastasis is angiogenesis. Now this is the process by which cells recruit blood vessels to grow close to them so that they have nutrients. Now there's a certain distance that cells can actually survive away from blood vessels because they need nutrients provided by the blood vessels in order to grow and to properly survive. When tumor cells start to grow and divide and become bigger and bigger, they suddenly seem to need more blood supply to keep those cells alive and to keep growing and dividing. And so they actually send out little chemical signals saying, recruiting blood vessels to come to that point and growing. So often you see, here's an example of a small tumor that's starting to develop. And you see it has a little bit of blood supplies to it, but as it grows more, it recruits even more arteries and veins to them so that the blood supply is always bringing nutrients to help those tumor cells grow and divide even more. And metastasis is facilitated by angiogenesis because it can pick up and move through the bloodstream. The cells can move through the bloodstream to other areas in the body. So I've looked at 5 different common factors with cancer, and you can see that there's many processes that are affected during the development of cancer. So when it comes to treating cancer, what many treatments that are currently available are for, they are aiming at treating features that are common for all forms of cancer. In other words, they want to suppress cell proliferation. They want to stop the cell from dividing and growing. And many common cancer treatments right now are aimed at doing this. Unfortunately, if you try to inhibit cell division, these treatments are not going to specifically be targeted only to cancer cells. They're also going to be targeted to any other cell that's actively dividing. So you're going to have ill effects in those cells as well. That's why individuals that are being treated for cancer often lose their hair, because those cells involved in the process are actively dividing. And so the cancer treatment also targets those cells. Radiation is also commonly used to treat cancer, and whether it's an indirect or a direct effect, either way it damages DNA enough in the cancer cells to stop them from growing and to therefore eliminate the growth of the tumor. Gleevec, shown here, this is the structure of Gleevec. This is a cancer treatment that actually has had very, very good success, and because it is targeted towards a protein called BCR-ABL. Now this is the result of a chromosomal translocation, actually, a fusion between two genes, and this results in a tyrosine kinase that is overly activated, and normally signals cell growth and differentiation. So when it's overly active, it helps to continue propagation of the cell. And so Gleevec is targeted towards inactivating BCR-ABL, and thus it prevents cell proliferation of the cancer cell. Gleevec is used for treating chronic myelogenous leukemia. And as I said, it inhibits cell proliferation and induces apoptosis of those cells that are affected. Now, while we're on the topic of cancer therapy, I'm going to get on my soapbox a little bit here and say one of the most important things to consider, especially now when you're fairly young, is the fact that there are some important steps you can take in your own life to help prevent the development of cancer. If you think about it, you don't want to be dependent upon the development of cancer drugs to treat your possible illnesses, when you can actually take small steps in your own life to help prevent this from occurring. And the book, your textbook, says that actually a half of all cancer deaths could be prevented. That's an estimation of how important it would be to play a role to take an active role in preventing these sort of things in your life. So, for example, some steps that could be taken are smoking. Quitting smoking is a great way to quickly decrease your chance of developing cancer. I realize it's a hard thing to do, but it's also very, very effective in helping preventing cancer. Sunlight exposure, putting sunscreen on when you go outside or when you're going to be out for a long period of time, it's really important in helping preventing exposure to UV light. And it's a small thing that may be an inconvenience now, but could save your skin in later life. Eating healthy, get your vegetables and your fruits. That's very important for getting all the important nutrients that can help prevent cancer. Minimize your exposure to chemicals that are known to be carcinogens, as well as microorganisms, specifically maybe some STDs that might contribute to cancer, like human papillomavirus. These simple steps in your life can actually help you later on if you think about it now. Yes, and they're also very important in preventing heart disease and actually an overall healthy lifestyle, and probably an improved quality of life as well. So it'd be nice someday if you could go in and, well, it wouldn't be nice if you have cancer, but suppose you do go in and it turns out you have cancer. It'd be wonderful if the doctor could simply take a screen of a cancer cell and say, well, it looks like this gene and this gene and this gene are involved in your cancer development. So we're going to take this drug, which is specifically designed to treat this gene, and target that. And thus we should be able to solve this problem real quick. That's not very feasible right now because we don't quite know enough and don't have the drugs available. But I think that actually is an idea that a lot of drug companies have, as well as a lot of cancer biologists. That's kind of a goal that I've heard many people suggest as we learn more about cancer development and as we discover what genes really do play a role in cancer. One thing that helps or that might actually contribute to this specific treatment of drugs is the idea of molecularly profiling cancer cells. So you can basically, this is an example of a DNA microarray, which has many genes, each little spot on here actually represents a gene from a cell. And you can actually profile which genes are activated, inactivated, and so on in a tumor cell. You can actually distinguish between different tumor cells if we can actually molecularly profile each of these different types of tumors. And this would be done by looking at levels of the concentrations of mRNA in these cells. Actually, it would be really useful for diagnosis as well to say, this is what's going wrong in the cell. And as I mentioned, could be used for specific treatment of your specific cancer or tumor cell that you have developed. So it's kind of an idea. This is definitely still something that's being developed and being used pretty widely now for cancer development and understanding the research to understand how cancer is affected and different kinds of cancer can be developed. Okay, so I'm going to actually take a break right here. And I brought along some note cards because I would actually like to get some feedback from you guys. As Dr. Fulton mentioned, I would someday like to have a teaching position at a college somewhere. And so I would like to get as much feedback as you would like to give me, whether it's positive or negative or whatever. If you have any comments, you think my lectures are too simple or too fast or too hard or whatever, I would greatly appreciate if you could just, sure, thanks. If you could just give me some feedback on just what you think. It would really help me out, and it would really give me some pointers on what I need to improve on when I'm lecturing. So if you want, go ahead and write down. If you want to put your name on, that will be kept on file. And if you don't want your comments associated with your name, then go ahead and use 2 cards or put your comments in somebody else's card or something. And whatever. So. Cards over here. All right. Did everybody get a card? The one other card? Okay, good. I'll just give you another minute or so to write down, and then I guess after class you could probably just leave your notes or your comments here at on the other table there. Right. I agree. Is it okay if I go ahead and move on, or do you need a few more minutes or another minute or so to finish up? Either way. Well, I'm just gonna continue on. If you want to continue writing your notes, that's fine. Or you can do it after class or whatever. But so getting back to cancer and how it is a genetic disease actually. there's a couple of questions that you might think as you consider the fact that cancer is a genetic disease. First of all, how do these alterations in genes arise? How do they occur, leading to oncogenes and tumor suppressor gene inactivation? Well, we're going to look in this chapter how DNA damage and mutations can arise, causing genetic alterations, developing to cancer development. And another question is, how does the cell deal with damage? When the damage is present in the cell, there actually are mechanisms to repair damage to help decrease the likelihood of mutations being introduced into your genes. And we're going to look at some of these repair mechanisms. And this is just a simplified example of what a repair mechanism might do, where you have, in pink, this is a circle, the circular object is supposed to represent a specific enzyme that recognizes a form of DNA damage. In blue here, this little pop out from the DNA structure in blue represents DNA damage. And there are enzymes that do specifically recognize specific types of DNA damage, and they can come in and also recruit other factors to repair the damage, basically remove it so that it's not there anymore in the DNA. And we're going to look at some of the major repair mechanisms in the cell. But let's start first with sources of DNA damage. How can damage actually be introduced into your DNA? Well, there's chemical agents that can do this. They're called mutagens because they often introduce mutations into the DNA. Ionizing radiation often causes breaks in the DNA that's really not well handled, and also can cause two mistakes in religating DNA or in trying to replicate the DNA. And then UV light, as I mentioned earlier, sunscreen is important because UV light can actually cause damage in the DNA. And one common form of the DNA damage that results from UV light exposure is called a thymine dimer. It's abbreviated a TT dimer often. And that actually cross-links two thymines that are adjacent to each other. So if you have DNA structure where the two nucleotides in a row are both thymines, what happens is UV induces this, I guess, it would be covalent bond formation between these two bases. You can see here, this is the linkage between the two thymines. And as a result, this kind of pulls the DNA, or those two nucleotides, a little bit closer to each other than normal, causing a distortion in the backbone of the DNA. Basically, there's a bend that's not normally there in the DNA backbone. And as I will remind you, we talked about DNA polymerases and how they have an active site that's very tight and only allows a certain geometry of base pairs. Well, when these thymine dimers are covalently cross-linked and this DNA backbone is altered, it actually alters the structure of DNA enough that it doesn't fit into the active site of DNA polymerases, and thus they can't replicate. The same thing goes for RNA polymerase for transcription. They cannot handle this distortion in the DNA. And thus it leaves the cell up to finding other methods by which to accommodate this distortion and this damage. And usually very often those methods are mutagenic, introducing mistakes in the DNA. Other environmental sources of DNA damage are chemical mutagens, also called carcinogens. And these cause mutations. For example, from smoking, benzoate-pyrene. Here, the structure of benzoate-pyrene. It's a chemical mutagen that comes from smoking, also comes from car exhaust, and it's a DNA base adduct. Basically, what it likes to do is grab onto DNA base and stick onto it with a covalent linkage. And again, this becomes a very bulky distortion on the DNA. And with that much bulk, the DNA polymerase, RNA polymerase, cannot handle that sort of template. And again, there's other processes that have to come in and account for it, leading to possible mutagenic effects. Another chemical mutagen is called aflatoxin, shown here. This is a chemical that's produced by mold, actually, and it can contaminate food. And if it is in the food that you eat, it's metabolized within the body to a species that's reactive. this also forms a DNA base adduct, grabs onto the DNA and links itself to it, and is not handled well during replication or transcription, again leading to potential problems with the DNA. Yes? Actually, it looks like they could get intercalated because of the base structure, but I think benzoate pyrene actually just forms an adduct on the side of a guanine. But I don't know about aflatoxin. Now, it also turns out that your own cellular metabolism could be damaging to your DNA. It can be harmful because often oxygen radicals are byproducts of the metabolic processes that you guys just learned about and probably learned a lot for your last test. Well, these radical oxygens have this free electron that are able to cause a lot of damage with the DNA and also other things that might be around, such as proteins. What it can do actually with guanine is form a specific form of damage called 8-oxoguanine. Basically, if you look here, this is the structure of a normal guanine. This is carbon number 8 right here. And thus, you add on this free radical oxygen reacts with it to put an oxygen with a double bond onto carbon number 8. So it's called 8-oxoguanine, and it's often abbreviated as 8-oxo-G. 8-oxoguanine is mutagenic because it actually forms different base pairs than guanine normally would. Now, it can form the correct Watson-Crick base pair between guanine and cytosine. So you can have an 8-oxo-G to C base pair, but it can also cause, if you actually rotate this 8-oxoguanine into a different conformation, it can base pair with an adenine as well. So it makes an incorrect base pair. So if you have a G to A, the next round of replication is going to result in the formation of a T to A base pair. And thus, you're going to have GC to AT changes in the DNA. And this causes a point mutation in the DNA. And if that's in an important gene, such as a proto-oncogene or a tumor suppressor gene, it might actually contribute to carcinogenesis. There's also forms of DNA damage that just spontaneously form. An example is an A-basic site. This is the result of hydrolysis of the glycosidic bond between the sugar and the base in the nucleotide. So what happens is this linkage between the sugar and base is hydrolyzed, resulting in the sugar and the phosphate being present on the DNA backbone, but there's no base present. So if this A-basic site is present in the DNA during replication, what's it going to base pair with? There's no base there as the template, so it's going to cause problems. Chances are well, first of all, DNA polymerase doesn't want to handle an A-basic site, but if it does, it has no template to use, so it's just likely to stick in any base, therefore leading to another point mutation in the DNA. Another example of spontaneously formed DNA damage is deamination of cytosine. This is an oxidative deamination, and it actually results in the formation of uracil. So cytosine, the structure of cytosine is shown here. Deamination refers to the fact that this amine group right here is removed from cytosine, and an oxygen replaces it. And that makes the structure of uracil. If you remember, uracil acts like thymine, right? So it's likely to base pair with A if it's present in the DNA. And then again, leading to mutations. One nice thing is that uracil doesn't belong in DNA. It belongs in RNA. So it's often detected and can be removed before this potential mutation might occur. But it does occasionally occur in the cell as well. So what happens if there's damage present in the DNA? I've mentioned that this DNA polymerase doesn't handle it very well. Neither does RNA polymerase. So if this damage isn't replaced, is not repaired, it can result in point mutations, insertions, deletions, basically some big alterations. And if it's in important genes, then it alters the activity of these encoded proteins, which are important for the cell cycle. If the damage is bad enough or serious enough, it actually halts the process of replication and transcription. And thus, it might induce apoptosis. If the cell is resistant to apoptotic signals, though, it might lead to transformation and lead to tumor development. As I mentioned earlier, thankfully, cells have DNA damage repair mechanisms that can avoid this. There are some simple repair mechanisms that require only one enzyme to accomplish the process, and then there's some other more complex ones that require multiple enzymes. We're going to start with the single enzyme repair mechanisms. Now, in bacteria, there's an enzyme called DNA photolyase, and this is an enzyme that's activated when the cell is exposed to UV. If you remember, I mentioned UV induces TT dimers, or thymine dimers. So right away, DNA photolyase is also activated to help repair those TT dimers. And by itself, it can basically remove the covalent linkage and leave the thymines undamaged. Another single enzyme repair mechanism is methyltransferase, and these are found in mammals. Now, guanine can also have damage on the oxygen, which is off of carbon 8. So here we have an oxygen that's hanging out. And an additional methyl group can be added onto this oxygen, forming O6-methylguanine. Methyltransferase is specifically designed to remove this methyl group from the DNA. And what it does is it transfers the methyl group to an amino acid within the protein, and thus repairs the guanine. But in the process, that addition of the methyl group to the enzyme inactivates the enzyme. So the cost of killing one of these enzymes compared to leaving the damage in the DNA, it sort of reflects how important it is to maintain the integrity of the DNA and to prevent damage from being present in nucleotides. And as I mentioned, there are complex repair mechanisms, and 3 of them that we're going to look at in this chapter are mismatch repair, base excision repair, and nucleotide excision repair. Each one of these involves a basic process of an enzyme that recognizes what's wrong with the DNA, binds to it, and then recruits maybe other enzymes to facilitate the process of repairing it. So we're going to start with the mismatch repair system. So if there's incorrect base pairs in the DNA, say an adenine to a guanine, there's this protein in bacteria, it's called mut-S. And this is basically what this does is as DNA is being synthesized, mut-S monitors the newly made DNA and specifically recognizes mismatches and binds to the DNA when it finds the mispairs. Here you have a picture of mut-S, and in pink and red is the DNA. And here's a side picture of it where you see the DNA is again present, and we have mut-S. I kind of think this top picture here looks like 2 hands kind of grabbing down and feeling along the DNA, trying to sense mismatches in the structure of the DNA. Once it binds, it causes a bend in the structure of the DNA. You might be able to see a little bit in this picture where it kind of kinks the DNA. And that signals to a nuclease, another enzyme, to come in and cut the DNA on the strand that has the mismatch. Now you might ask, how does the nuclease know which strand has the mismatch when mut-S is bound to a double helix? Well, if you remember, DNA is normally methylated in cells, bacteria, and so newly replicated DNA is not going to be methylated yet. So the nuclease recognizes unmethylated DNA and cuts that strand specifically so that the DNA helix can be unwound and basically the defective DNA is removed and replication can continue over that area again and thus hopefully correctly replicate, this time leaving undamaged or no mistakes left in the DNA. Now that was mismatch repair system. And I think I'm going to stop here. On Monday, we'll move on with basic excision repair. Oh yeah, and if you have notes or comments that you left on note cards, I'd appreciate if you could leave them somewhere at one of these tables. I really like studying DNA damage.
Exactly what I was curious was what was defect that causes cancer. Right, yeah. And they just didn't show me a lot of